And without any hiccups in timings, I'll directly go to my presentation. And students, before I go to presentation mode, one short note to you people. It's a vast subject, immunology. And of course, I shall be opening your innings, and there are other dignitaries to follow me. This subject, when you have to study, you have to do it in conjugation with other areas, such as molecular biology, physiology, cell biology. Everything put together is immunology, in fact. Okay. So you just cannot stick around immunology as a separate entity. So with this note, I'll give me a second, I'll go to presentation mode. I hope my first screen is visible to everyone.
Yes, sir.
I have entitled my talk, Important Concepts for Understanding Immunology. This was done deliberately in consultation with Gujarat State Biotechnology Mission, because this subject is introduced for the first time in this e-crash course, initiative from GSBTM. My sound a bit weird, but I can't help it. Over the years, this has been the traditional way of introducing immunology to my subjects. So please excuse me, it sounds weird. Concept which I would like to give in a nutshell. On the left slide, you have traditional Indian cuisine, the pani puris. Often in a joking way, it is said, Mamma's handmade pani puris do not taste that good, but roadside pani puris are tastier. And one of the reason is, on a lighter note, that Mamma makes your pani puri at home, it may not have the pathogens. So, but here for my example, I am taking your roadside content, where certain amount of microbial flora will be always present. And we take a hypothetical situation that suppose this pani puri is laden with some of the called as GI tract specific microorganisms. What would happen is this animated GIF, within a couple of hours, that is the shortest duration, to several days later, maybe a day later, you will rush from left side of the PowerPoint slide towards the right side. Essentially, this concepts builds in your immunology that when I have to define immunity, it's the state of protection, and it's the bolus of infection multiplied virulence divided by immunity. Bolus of infection, how much microorganism is present in the content that you are taking. So that is bolus of infection. So how much microorganism is there in this content, that is your bolus of infection, multiplied by virulence. Yeah, you have microorganisms, but how virulent are these microorganisms? If it is lactobacillus, of course, it's non-pathogenic, so the virulence won't be there. Again, for the virulent microorganism, how virulent, what strength, what is the ability to cause pathogenicity, that is another factor, divided by your immunity. So when I say divided by your immunity, how powerful is your immune system, which can counteract the effect of these microbial agents that have breached the immune defense and has entered your system. So if your if your immunity in general is powerful enough, you can avoid the right side of the PowerPoint slide. And that is why whenever I give example, you have a subject who has consumed 200 pani puris, but still that person will be the first one to be present in the lecture. There might be others who might not have consumed might have consumed only 10 pani puris, but still that person would be absent from the next day's lecture. So this is all about the introductory aspects that I am going to give. So my next segment, I would move to the historical perspective of the subject, and that is what has been assigned to me. So when you talk about the historical perspective of immunology, the first question that comes in our mind, what is immunity? What is immunity? Immunity is a state of protection against foreign pathogen. This term foreign is very important because substances which are not in our system, the foreign pathogen or substances, those foreign pathogens or substances in technical language, we refer to them as antigen. I would restrict this to my next segment of the talk, which begins from 11:30. There I will be talking about the aliens in greater detail. But how this term came into existence? This term has Latin origin. This is from the word immunis. The word immunis means exempt. Just like suppose you have appeared for 4 examination papers, you have cleared 3 papers, but you have got 80 KT in one of the paper. So you are exempted from appearing for all those papers that you have already passed. That is exemption. So immunis means exempt, and it is the source of the English word immunity. Now, though immunology is a new subject, but the observation or the concept of immunity is not known, not new. In fact, if I tell you, the literature says that observation of immunity goes back over 2000 years. It was 2000 years ago, there was an ancient historian in as early as 430 BC. He observed that there was a great plague in Athens where all those who recovered from plague, they could effectively nurse the ill persons. So there was a reasoning that those who came in contact with plague but later on recovered, they might have developed immunity. But then the subject was not born, but the concept was very much present some 2000 years ago. So that was the primary mention of immunity. Now, when we move towards immunology, we move towards the subject, it was 1718. 1718, you have Lady Montague, the famous and was variolation. And when she returned to Britain, she introduced it in her country. And not only that, to promote this concept of variolation, she got her own children variolated. And this particular right side panel, what you see, this is a memorial burst of Lady Montague. Her contribution toward the subject, even without being a faculty of immunology, is enormous because she was the one who popularized immunology in a mainstream society. So that's how we move with immunology. Then came the ultimate father of the subject. I hope from the figure itself, you can very well know that this is the first entry of Professor Dr. Edward Jenner. So, can we generate immunity without inducing disease? The answer is yes. These days it is much in news. You can do it through vaccination. Now, what happens in vaccination? Vaccination prepares the immune system to eradicate an infectious agent even before it causes disease. I hope many of you or many from the fraternity, especially in India, who are 18 plus, they might have taken at least one dose of COVID-19 vaccine. So vaccination prepares the immune system to eradicate infectious agent even before it causes a disease. This concept of vaccination has saved so many lives. Many disease has been stamped out of this world just because of vaccination. And one such disease, of course, is smallpox. So some of the classical examples of vaccination: rabies vaccine, the eradication of smallpox, and these days, the childhood diarrheal death has been stamped out because of rampant use of the rotavirus vaccine. It's now in government schedule of vaccination. Earlier, it was an optional one. Then you have DPT, diphtheria, pertussis, and tetanus. You have MMR, measles, mumps, and rubella. So Mission Indradhanush program is there from Government of India for vaccination. And now, of course, Covishield and Covaxin are the two indigenous variants of vaccines that are administered in our country, of which Covaxin, of course, is completely indigenous from Serum Institute of India. Recently, you might have read in news, a manufacturing hub has been started in our neighboring district of Ankleshwar in Gujarat. So that was the initial perspective of immunology. Then comes the famous person from microbiology, Louis Pasteur. 1879, he discovered that old bacterial culture of Pasteurella lost virulence. So whenever you are keeping the culture for extended duration of time, the bacterial culture loses virulence. He referred to the weakened culture a vaccine in honor of Jenner. So this is what you try to understand. One aspect that, so Edward Jenner was never the person who coined the term vaccine. Rather, it was Louis Pasteur. In memory of Edward Jenner, he coined the term vaccine. So in MCQ, in IIT JAM or JNU CB, if the question is asked, who is the one who coined the term vaccine, please don't write Edward Jenner. Your answer should be Louis Pasteur. Then came 1881. He applied the concept of weakened culture to some of the popular diseases existing at that time. And this diagram or this popular painting, you can see Louis Pasteur applying the concept of vaccine to anthrax as well as rabies. In those days, many sheep used to die because of anthrax, and he was the one who pioneered both rabies as well as anthrax vaccine. That was Louis Pasteur. Then comes Professor Emil von Behring. He was the one who won the Nobel Prize in Physiology and Medicine 1901. In fact, someone asked you who was the first immunologist to ever win the Nobel Prize in Physiology and Medicine. It was in 1901 that the first Nobel Prize went to this subject, that is immunology. And you might be aware the subject immunology and its discipline has won Nobel Prize several times. You had autophagy, you have innate immunity, Professor Jules Hoffman, right? So many concept of immunology Nobel Prize has been there. And Emil von Behring won the Nobel Prize for his seminal work on serum therapy, especially in application against diphtheria. So in application against diphtheria, the Nobel Prize. Then comes the famous pair, Elie Metchnikoff and Paul Ehrlich. It's a different thing that they had a bit of strained relation in the later part of the life. Of course, you can read from Nobel Foundation's website. So Elie Metchnikoff discovered phagocytosis by macrophages I am highlighting the important areas because these are some of the questions. As a critical host defense mechanism, and thus he is considered as the father of cellular innate immunity. So, in fact, Mechnikov started the concept of innate immunity and hence is called as the father of cellular innate immunity. Paul Ehrlich worked on toxin antitoxin reaction, and he is also the one who pioneered the famous side chain theory, okay, for disease manifestation. Now, based on the initial first segment of the talk spanning over one quarter of the allocated time, I would like to ask my students the first question. Please wait for a while. Please don't answer right now. Let me visualize the chat box. In the meanwhile, I'll read out the question, which is the best definition of immunity. There are 5 options, and you have to hit the right option in your chat box so that though our lecture is in virtual mode, we will get some fun as if it's a live interaction that's taking place. Okay, I believe the students are on YouTube live, so I might not be able to see their direct interaction at the moment. I don't know whether if other students on Google Meet, then of course you can hit the chat box. Yes. So which is the best definition? Yeah, my chat box is ready. I can see your answers
B
right
option.
now.
Sir, chat box is ready and students can post messages in chat box.
Fair enough. Well, the messages have already come. Option B, majority of the students are going, and yes, we would go with option B. That's the right answer. The state of being resistant to reinfection with a pathogen, that's the best definition of immunity in the perspective. This, of course, is not directly related to my talk, but nevertheless, since you have already been introduced to the subject, so molecules directly involved in NK cell mediated, natural killer cell mediated killing involved muramyl dipeptide, granzyme A and B, the complement system, interferon gamma, and complement C3b. I would like to add a note that complement system, of course, is not taught. It will come in succeeding part. But in innate immunity, you have a cell type called as natural killer cell, and these are MHC independent, that you don't require MHC for its activation. So molecules involved in NK cell mediated killing involves, yes, anyone can guess in the chat box. Katara Pranav is going with D, of course, is not the right answer. Tejas Vaghela is also going with D. No, it's not the right answer. Without wasting time, I'll give the right answer. The right answer is granzyme A and B. Remember, directly involved in natural killer cell mediated immune response. And of course, you also have the perforin granzyme pathway where also granzyme A and B is involved. Okay, we move to the next one. Now we come to overview of immune system. When you talk about the immune system is divided into 2 parts: innate component, which is non-specific, also called as the first line of defense, and an adaptive component, which is specific, also called as the second line of defense. This adaptive component or the second line of defense protects us against re-exposure or when you suffer from the same infection a second time. Again, innate and adaptive immunity has two components, a cellular component and a humoral component. Both cellular and the humoral component, they will interact with each other. And again, both innate as well as the adaptive immunity, they also work in a coordinated fashion. Initial responders are the innate immune system, but the fine touch or fine tuning or the late responders are the adaptive immune system. So you have an interaction not only between the cellular and the humoral component of immune system, but also between the innate and the adaptive immune system, wherein the adaptive immune system is also the second line of defense. Now, in this next segment, we are still into the overview of immune system. In the next segment, what we would do, we would focus on non-specific defenses and specific defenses. When you talk about non-specific defenses, we essentially talk about innate immunity. Innate immunity being the first line of defense, and innate immunity also has a second line of defense. The first line of defense consists of skin, also called as saran wrap of your body, because whenever your skin is breached, your skin has injury, you have lacerations, cut, the pathogen can easily enter. The mucous membrane and the secretions from skin and mucous membrane. Psoriasin is one such secretion which prevents colonization of E. coli in your skin. Second line of defense, you have phagocytic leukocytes. Leukocyte, as the name suggests, leukos is white, cytos is cell. So white blood corpuscles or leukocytes. Second line of defense also consists of antimicrobial peptides. These are natural peptides present in component of the innate immune system. The inflammatory response, which wards off the pathogen, and fever. Fever is also a component of innate immunity. Fever per se is not bad. Mild amount of fever is always good because it's a component of innate immunity. Why you get fever is because you get fever to essentially ward off your pathogen using a raised body temperature. You have specific defenses, adaptive immunity. Adaptive immunity happens to be your third line of defense. An adaptive immune system has lymphocytes, the antibodies, and the memory cells. Now we have humoral immunity. Can anyone unmute and speak? Which type of immune cell comes in your mind when we think or talk about humoral immunity? Anyone?
B cells.
Exactly, B cells, that's right. Humoral immunity combats pathogen via antibodies. And whenever you talk about antibodies, you have antibodies produced by B cells. Of course, when you talk about B cells are naive B cells. Once the B cells differentiate, they proliferate, they become a plasma cell. The plasma cells are the ones which are called as the effector B cell, and the effector B cells produce the antibodies. And you have two types of memory B cell and the effector B cells. Memory B cells will remain in circulation. So the second time you suffer from the same disease, it renders protection. Interestingly, antibodies can be transferred between individuals. And when antibodies are transferred between individuals, they provide passive immunity. So while passive immunity can be transferred by transferring antibody between two individuals, you get passive immunity. We were talking about immunologic memory. Immunologic memory is a component of the adaptive immune system. So let's see what happens in diagrammatic mode. On x-axis or abscissa, we have time in days. Y-axis or ordinate, we have magnitude of immune response. We have two color shading that we have used. One is pale blue, another is slightly darker or sky blue. If you see the pale blue is your innate immune system. If you see innate immune system, if you see the y-axis of innate immune system is similar, no change. Whereas if you see the peaks of adaptive immune system, when you are challenging with antigen A, the amplitude is less, but you repeat the same antigen A, the amplitude increases. This much is the increase in the amplitude or the peak. So what happens for innate immunity, whether it was the first time you are injecting the pathogen or the antigen, or you are injecting after 14 days, it's one and the same response. But when it comes to the adaptive immune system, what happens is you have the initial response, the amplitude is less. Later on, you repeat the same antigen, the amplitude increases. So this is the secondary immune response. And again, the duration of time taken in secondary immune response is much less as compared to primary immune response. Before I go to the next segment, can I have a bit of interaction? Would any student tell me what might be the reason that the secondary immune response is always duration-wise shorter than the primary immune response? What might be the reason? Anyone? If you are aware, you can just unmute and speak. If not, we go to the... Yes, please.
सर सेकेंडरी रिस्पॉन्स मेमोरी बेस्ड होता है, मतलब उसकी मेमोरी हमारे बॉडी के सेल्स के पास पहले से है, और जो फर्स्ट है उसमें इंटरेक्शन करके आइडेंटिफाई करना है कि कौन सा एंटीजन हुआ है और उसके लिए कौन सा रिस्पॉन्स देना है, और फिर बी सेल प्रोड्यूस होंगे, बी सेल प्लाज्मा, फिर
Exactly, I got the answer. Sorry for interruption. Actually, what the last part is more important, since for production of antibody, you have to convert, or the naive B cell has to get converted. The naive B cell, you have a naive B cell, I will put it as N. The naive B cell has to undergo transformation into a effector B cell. Only then the plasma cell or effector B cell will produce antibody. In primary immune response, when you talk about the primary immune response, the primary anti-A response is much less. But the same antigen you are injecting again, the secondary response is much higher. As you rightly said, the memory cells are already there, so they will directly produce the effector without having to wait. But if you see the antigen B was not injected in the primary response. It is directly injected new. Therefore, the primary anti-B response is very similar to anti-A response. You see the both amplitude are matching. So therefore, this proves us that you have secondary immune response where the B cell proliferation into effector B cell. Effector B cells secrete two types of cells, the immunoglobulins or the antibodies, and the memory B cells, which will remain in circulation. Now, before I come to the next slide, once again, one more round of question to my students. Can anyone or would anyone like to tell me which is the first immunoglobulin that is formed?
IgM.
IgM is the first immunoglobulin that is formed, quite right. So if you see the primary immune response, IgM, And if it is an innate immune response, IgM titer remains the same. But if you see the secondary immune response, if you see the IgM titer is less, whereas the titer of immunoglobulin G is significantly elevated. What might have prompted this immunoglobulin M to become immunoglobulin G? Of course, that part of the talk will be deliberated by Dr. Amit Balani in today itself. So he will be talking about immunoglobulin gene rearrangement, and at that time he would talk about what is class switching. Essentially, when one immunoglobulin switches its heavy chain class and becomes another immunoglobulin, that is called as class switching. Here also, immunoglobulin M switches its heavy chain or switches its class, and it becomes immunoglobulin G, which is another class of immunoglobulin. So someone asks you an MCQ, which is the first immunoglobulin in primary immune response, your answer should be immunoglobulin M. Which is the immunoglobulin in secondary immune response, the answer should be immunoglobulin G. If you talk about cell-mediated immunity, the cell-mediated immunity involves primary T lymphocytes. These primary T lymphocytes can eradicate pathogen and they can clear infected self cell. Once the infected self cells are cleared, they in turn will help other cells to induce immunity. Based on that, we come to some of the questions from IIT JAM 2019. Following statements are true regarding primary immune response. One of the following is not true, except, that means everything is true except which one is not true. I am again ready on my chat box. You can just answer. It occurs on first exposure to antigen. It is applicable to adaptive as well as innate immunity. It does not use immunologic memory. It is applicable to both humoral and cell-mediated immunity. What will be the answer? Manthan Choksi is going with C. Katara Pranav is also going with C. Sanidhya Jude and Nancy Mehta is most of them. So far, whatever answer I've got is all going with option C. True with respect to primary immune response, except, okay, it occurs on first exposure to antigen. Yes, that is correct. So you cannot cross it off. It does not use immunologic memory. Yes, that is also correct. Primary immune response do not use immunologic memory. It is only the secondary immune response that use immunologic memory. Third option, it is applicable to both humoral and cell-mediated immunity. Yes, that is also true. Humoral and cell-mediated immunity both can bring about the primary immune response. So that also holds true. And so finally, the answer would be option B. It is applicable to adaptive as well as innate immunity. That is the correct option for one of the following statements are not true regarding primary immune response. Now we come to the very important concept called as clonal selection. You have a stem cell, hematopoietic stem cell undergoes gene rearrangement. It becomes a mature B cell. Mature B cell will secrete immunoglobulins. These immunoglobulins can come in contact with antigen. Once it comes in closer contact with antigen, then begins the antigen-dependent proliferation of mature B cell. Now, what do we mean by antigen-dependent proliferation? If you see the color code, the antigen 2 interacts with the mature B cell type 2. So, whichever clones will be produced, all will be against antigen 2. If it happens to be antigen 3, all clones will be for antigen 3. So this concept you need to remember. And after a particular antigen has proliferated a certain number of B cells, those B cells will transform into a memory B cell and a plasma cell. And just now I said earlier that plasma cells are the ones which are going to secrete immunoglobulins or antibodies. One of the reasons why compared to a naive B cell, the size of plasma B cell is considerably bigger, because plasma B cell has lots of cell organelles to take care of its survival as well as for its synthesis. Can be due to each cell having many copies of receptor on the surface type of antigen. Either they will be having many copies of receptor on the surface, or they can bind to one specific type of antigen. When the B or T cell interacts with specific antigen, it is selected, and this selection and further activation is called as antigen dependent T cell or B cell activation. So each clone is reactive against antigen that initially surrounded it. Why each clone is reactive against antigen that is initially stimulated? Because when a narrow range of antigen is stimulated, it will lead to a clone that is reactive against the antigen initially stimulated. Again, to go back to the diagram, if it was antigen 2, then this pink mature B cell will develop. If it is antigen against the yellow would develop into the lineage. If it is blue, the blue ones would develop, or if it is the green ones would develop. So that's antigen dependent proliferation and differentiation into plasma and memory cells. Part of them will become plasma cells and part of them would become the memory cells. Now the question is, which of the following is not a principle of clonal selection theory? It's slightly tricky. Please read all the options and only then you answer. Anyone? The correct answer is option C. Receptors with different specificities are produced at the offsprings of activated lymphocytes. And if you go with the diagram that I explained, one antigen will stimulate a specific type of antibody against the same antigen. So when you have receptors with different specificities, right, so they will be produced as the offspring of activated lymphocyte. This is, of course, not a principle of clonal selection. So that's the right answer. Now I will connect the two components of the immune system, the humoral component, the humoral response or B lymphocytes, with the cell-mediated response or T lymphocytes. The B lymphocytes will encounter antigen, just now what we were seeing or what we were understanding. Once the antigen is encountered, it will become B cell proliferation, activation would take place. The B cell will produce antibody, activated B cell will produce antibody, and these antibodies will lead to antigen elimination. But when you talk about the cell-mediated response or T lymphocytes, you have T cell receptor, just like the B cell. The T cell receptor will also interact with antigen, and once the antigen interacts, it will lead to killing of infected cell. However, in cytokine secretion some of the cytokines help in the T cell receptor to become the activated T cell, which will kill the neighboring cell or the target cell. Now, in this diagram, two things are to be seen. First, the cytokine from the T cell. This cytokine, if you see the arrows, this cytokine can help the naive T cell to become activated T cell. Again, the same cytokine is also helping the B cell to develop. And it is for this reason that one type of B cell development, we call it as TI, thymic independent B cell development. And another type of the B cell development is called as, one is the TI, T independent, another is the TD, which is also called as thymus dependent. Of course, thymus dependent, the quality of antibody is much better as compared to thymus independent mice. Nevertheless, it's possible. So which of the following types of immune system cells are known as inflammatory cells and recruit to infected areas in large number where they engulf bacteria and other microorganisms? Any of the four options that you can tell. Prachi, your answer is B. Can if you have read the question, can you hit the chat box, students? Okay, answers have started coming. It is so which of the following types of immune system cells are known as inflammatory cells and are recruited to infected areas in large number? This is called as neutrophils. Neutrophils is the one. Now we move on to the next segment. In this segment, we would talk about the difference between innate and adaptive immunity. As the diagram suggests, the diagram of hare and the tortoise, innate immunity is quite rapid as the hare, but non-specialized. Adaptive immunity might be slow, just as slow as the turtle, but then it is very determined that it has to reach the final spot. Now, how do you compare the innate and the adaptive immunity? What is the ideal comparison that you can do? In response to pathogen, vertebrate immune system are two interconnected systems. One is innate immunity, another is adaptive immunity. Let's see the difference. Response time in innate immunity is minutes to hours, and adaptive immunity, it will take place within days. Specificity, innate immunity is limited and fixed. Adaptive immunity is highly diverse. Each time it encounters an antigen, it will trigger an anti-immunologic response mediated by the course of immune response that initially set in. Now, response to repeat infection, innate, every time it is going to be same, right? Just like an अंगूठा छाप इंसान, कितना भी सिखाओ फिर भी वही करेगा. Then, in case of adaptive immune response, more rapid and effective or subsequent exposure might be there that might be responsible for adaptive immune response. you talk barriers, only then it can enter. And this is how the anatomic barrier to infection is a protection in innate immunity. Physical barriers, epithelial surface, keratin layer of intact skin, mechanical respiratory cilia, the flushing action of tears and saliva. We also have the competing with pathogenic bacteria for micronutrients. When you talk about the inhibition of bacteria, it's always the breakdown of cell wall bacteria, or it can destabilize the bacterial membrane. It prevents the growth of bacteria, and it also ingests and destroys microbes. You talk about the biological action of this. The normal flora of skin, throat, colon, the vagina, and the gastrointestinal tract are the targets of the biological barriers. So prevent the colonization of pathogenic bacteria by secreting toxic substances and the competing with putative bacteria for nutrients. So students, what I want to tell in very nutshell that there are anatomic barriers to infection. You have mechanical barriers, you have the physical barriers, you have the chemical barriers, and the biological barriers. All these anatomic barriers to infection prevent the pathogen from directly entering the cell. Only when there's a breach in this design, then the infection can easily set in. Otherwise, it is always subjected to protection. Amongst that, the low pH of sweat and the gastric glands of the stomach, the lysozyme present in tears and other secretions. So whenever you are crying, ideally you are solving your eye infection by crying, because when you cry, the flushing action of tears would be there, which is bacteriostatic. Likewise, when you spit, one of the ways phagocytosis, which won Mechnikov and Paul Ehrlich a Nobel Prize, is phagocytosis. What's this phagocytosis? It's described as engulfment and internalization of materials such as microbiomes for their clearance and destruction. Now, phagocytosis, I always tell in my language as the hug of death. So this is a pathogen. This is a pathogen. Pseudopodia projecting the pathogen, and once the pathogen is internalized, it is subjected to the phagocytosis, and this phagocytosis will lead to the phagocytosis. You have cell wall component, the complex carbohydrates such as mannans and glucans, the lipopolysaccharides, the other lipid containing molecules, and peptidoglycan. Students, when you talk about PAMs, try to understand PAM stands for pathogen associated molecular patterns. Initially, scientists told it as, immunologists called it as PAMs, pathogen associated molecular patterns. But now it is very well known that every pathogen may not be, or every microorganism may not be a pathogen. Lactobacillus, it is a microorganism, but is it a pathogen? No, it's not a pathogen. So we have given a general term, that is MAMS. MAMS stands for microbe associated, okay, the PAMS is pathogen associated molecular patterns. MAMS is microbe associated molecular patterns, that is MAMS. What comes in your mind when you just imagine this cheese burst pizza? On cheese burst pizza, you have lots of toppings. You have the jalapenos, you have the chili flakes, you have the oreganos. So ultimately it will feel like salivation in your mouth. So the concept I would like to help is opsonization. Just like when you are adding all these toppings to your pizza, it becomes tasty. Likewise, the microorganism can be coated, and once it is coated, it is subjected to the phagocytosis. It enhances phagocytosis. Enhancement of phagocytosis by coating with the particles is called as opsonization. Opsonization means to make tasty. And once the microorganism is made opsonizable by soluble phagocytosis enhancing protein, the soluble pattern recognition proteins will make it capable of opsonization. Let's have some question before we end the session. Which of the following statements is true for innate immune system? Its response generally involves the production of antibodies. Its response generally involves macrophages. It provides tailored response for specific pathogen. It is responsible for medical efficacy of vaccine. What would be your answer? Hit the chat box. Well, people are going with option A, B, D. All options people are clicking. So first of all, you rule out some of the options. Okay, its response generally involves production of antibodies. Okay, innate immune system, yes, it does produce antibodies, but generally it's not the only one that it just produces antibodies. Its response generally involves macrophages. It provides tailored response, and it's responsible for medical efficacy. The correct answer, just now we talked about the Nobel laureate Meknoff. You have its response generally involves macrophages. So most of you, that is Muskan Singh, Katara Pranav, and Mirchandani Ishita, they all have got the correct answer. Option B is the right answer. Phagocytosis is carried out by cells of the adaptive immune response, is restricted to macrophages, important in bacterial infection, and the process does not involve energy, results in division of the cell. What would be the right answer? Okay, people are taking time to think. Yes, Sanidhya has gone with C. Well, most of them are going with C. Yes, phagocytosis is important for the bacterial infections. That's right. Which of the following statement best differentiates innate and adaptive immune response? The options are slightly lengthier. You patiently read it, and after that you hit the chat box. So by the time you hit the chat box, so you can just think. Well, yeah, just a minute. I'm reading your answer in the chat box, and if you cannot see my presentation, don't worry. I have deliberately logged itself out because just now I'm just about to come to the final part of my presentation. Okay. Well, the correct answer, of course, is option C. It's important for bacterial infection. Now I move to the last segment of my today's talk in this introductory of immunology. When you talk about innate immunity, we cannot leave this topic of inflammation. Inflammation is from the Latin root inflammatio, which means to set on fire. So recently we had COVID cases all across the world, the pandemic, and there we talked about lots of inflammation. No? We talked about that COVID kills because of inflammation. So whenever you talk about inflammation, we have a pro-inflammatory cytokines and anti-inflammatory cytokines. Whenever you have inflammation, the pro-inflammatory cytokines, the level goes up. The anti-inflammatory cytokine, the level goes down. Okay. So we talk about inflammation. The fundamental host response to injury is inflammation. What inflammation does, it sends activated immune cells to the site. It sequesters pathogen. That is, pathogen will be engaged with the inflammatory agents. It limits damage. Whatever damage has happened, it's fine. It won't cause further damage. That is, limits damage. And the best part, inflammation also initiates repair. Now, inflammation is of two types, acute inflammation and a chronic inflammation. Acute inflammation is a response to tissue damage, and chronic inflammation, where is a long-term basis such as arthritis and wasting of tissues. It has four classical signs: calor, which is heat; dolor, pain; rubor, redness; and tumor, which is swelling. I have given a certain funky diagram so that you can associate heat, pain, redness, and swelling with the associate clicks that I have given. To talk about inflammation, initially there is a vasodilation, edema, and extravasation. In vasodilation, what happens is there is a rise in blood volume and there is a heatening and reddening effect. In edema, as the name suggests, there is a leakage of fluid from post-capillary venules. This leakage will adhere to the endothelial cells in the inflamed region. Because of action of vasodilation and edema, the capillary walls become more permeable, and this leads to the escape of fluid through the capillary space. So I will just introduce you to the diagrammatic effect. There is a breach in the immune system, the anatomic barriers such as skin, the bacteria enters. Initially, there is vasodilation, increase in the blood vessel diameter. It was the diameter of this. It increases the diameter, more blood, more mediators. After that, the endothelial junctions will become permeable. It results in edema. The endothelial junctions become permeable, this part. Finally, the neutrophils will extravasate. It will squeeze through the gaps, and after extravasation, the phagocytes will reach the site of infection and it will carry out phagocytosis, that is the antibacterial action. Inflammation is always a double-edged sword. For immune system, it is beneficial because infection is kept under control. It is bad because inflammation can also lead to wasting away of tissues. And that is why we always take NSAIDs, non-steroidal anti-inflammatory drugs, to prevent inflammation. Again, inflammation is of two types, the acute inflammation and the chronic inflammation. Okay. So I know it's not enough for entrance examination, but what I did, I just introduced you to the subject of immunology. I gave you the concepts of innate versus adaptive immunity. What are the components of the innate immune system? How innate immune system is the first line of defense. Innate immune system coordinates with the adaptive immune system to help in the entire array of reactions leading to limiting of pathogens. Now the subject is wide open. You have my next session where I will talk about the alien that has entered. This is the alien that has entered, correct? So we will talk about antigen, we will talk about antibodies. Then we have Dr. Hardik, who will be talking about the antigen-antibody interactions. And finally, today's session will be wrapped up by the veteran, the person who is long is lymphocyte receptor gene rearrangement. He will be talking about how immunoglobulins rearrange to functional aspect of immune system. So with this, I would like to end my first session of today's introductory immunology. Having said about immunology, it would be unfair without giving tribute to my guru, Professor A.V. Ramchandran. Way back in year 2000, or maybe before that, he was the person who introduced me to immunology. And of course, this is my student, Miss Megha Dave. So it's almost 3 generations you can talk. So thank you very much for your patience. You can just wait as per the instruction of the panel members of the host institution. And thank you very much. If you have any queries, the session is open for discussion.
सर
Yes, please.
सर आपकी जो फर्स्ट प्रेजेंटेशन थी ना उसमें थर्ड क्वेश्चन आप निकाल सकते हैं एक बार, एक डाउट है मतलब.
Okay, fine. Third question, right? Third question, okay. Following statements are true regarding primary immune response except